Hello, welcome to our presentation. My name is Ashley Mack.
And I'm Austin Smith, and today we're going to talk about what Hox genes are, and specifically how these genes present themselves in Drosophila.
Also known as fruit flies. These are going to be your learning objectives for this presentation. We're going to focus on, number 1, your understanding of what a Hox gene is and what its purpose is. 2, knowing the patterns of Hox gene activity. And 3, understanding what homeotic transformations are and making sure that you're able to recall the few homeotic transformations of Drosophila that we will go through later.
So some of you might be wondering what a Hox gene actually is. So a Hox gene is more formally known as a homeotic gene, and the purpose of this gene is to regulate and determine what each segment of the organism will turn out to be.
Hox genes are located at different points along the chromosome. Their locations on the chromosome relate to their segmental presentation within the organism. For example, the gene at the most 3' end of the chromosome corresponds to the head segment of the organism. Likewise, the gene at the most 5' end of the chromosome corresponds to the posterior or butt region of the organism. Each and every cell is capable of creating any structure of the fruit fly, as each cell contains the DNA that holds all of the genes. However, the Hox genes are what provide the roadmap to allow these cells to create the structures necessary to divide the fly into the proper segments. We learned in general genetics that an organism has many different types of genes that regulate the division of the body. The more general genes are the gap genes and parallel genes. These genes and their activation and expression interact, and as a result, they can activate or repress the expression of Hox genes.
Here is a pictorial depiction of how the location of each Hox gene along the chromosome translates to the physical segmentation of the Drosophila. As you can see in the image right here to the left, the darker region, which is depicted with the brown color, codes for the head. The middle segments, which are depicted by the orange color, will code for the thorax, and the tail region, which is depicted with the yellow color, will code for the abdomen. Here's a quick video of Hox genes in actions as you can see the complete development of the Drosophila and the end result of the different body segments.
The Hox gene patterns are broken up into separate groups, the Antennapedia cluster and the bithorax cluster. These groups are named as they relate to the issues that can arise when these genes are disrupted. The Antennapedia cluster includes 4 genes. The first is lab or labial, the second is DFD or deformed, the next is SCR or sex combs reduced, and the last is ANTP or Antennapedia. The labial and deformed genes specify the head sector, and the SCR and ANTP genes contribute to the thorax segments. The bithorax cluster includes 3 genes, the UBX, or ultrabithorax gene, the ABDA and ABDB, or abdominal A and abdominal B genes. The UBX gene is required for identity of the third thoracic segment, while the ABDA and B are responsible for the segmental identities of abdominal segments.
Each of the proteins produced from Hox genes contains a homeodomain, consisting of 60 amino acids encoded by the 180 nucleotide homeobox. This allows the protein to bind directly to DNA as a transcription factor, activating other genes, and thereby directing pattern formation in the developing embryo.
This picture shows the 2 groups of Hox genes, the Antennapedia and the bithorax, and their positions in the Drosophila in both the adult and the embryo. As you can see, the labial, depicted in the red, and the deformed genes, depicted in purple, are expressed in the head. Sex combs reduced, which is the yellow, and the Antennapedia, which is the brown, are shown in the first 2 thoracic segments. The ultrabithorax gene, which is the teal color, can be seen in the third thoracic segment, and the abdominal A and B genes, which are the last blue and green, can be seen in the abdominal segments.
A homeotic transformation occurs when the regulation of the Hox genes becomes disrupted and segmental changes or mutations occur. The two most common transformations in Drosophila are antennapedia and bithorax. Antennapedia is the most common transformation. What occurs with this transformation is that the coating for the antenna gets disrupted and legs form in place of where normal antenna should form. Bithorax is the second most common transformation. What occurs is that the coating for the posterior segment of the fruit fly is disrupted, and a second thorax segment forms in its place. As a result, this Drosophila exhibits 2 thorax segments and 4 wings.
Here's a great depiction of the antennapedia transformation. As you can see, the antennae on the left are small structures that are located on the face. The antennae have a wide variety of sensory functions, including olfactory, or smell, and auditory, or hearing. When the coding for these genes get interrupted and the legs form instead, as you can see on the right image, this rids the Drosophila of being able to obtain that sensory data that it otherwise would have.
Here is a great depiction of the bithorax transformation. As you can see on the left, a wild-type fly exhibits 1 pair of wings and a pair of what are called halteres. The function of these halteres are to aid in balance and guidance for the Drosophila and its body position while flying. While the bithorax mutation, as seen on the right, has a repeat of the thorax, resulting in a second pair of wings, and as a result, the possibility of reduced balance.
Alright, so now that we've learned all about the Drosophila, it's time to do a quick recap.
First off, a Hox gene is a homeotic gene that decides the segmental identity of an organism, or in our case, the Drosophila or fruit fly.
And the patterns of activity are divided into 2 clusters, the antennapedia and the bithorax cluster, resulting in a total of 7 Hox genes to determine the segments in the Drosophila, with the first 4 genes in the antennapedia cluster coding for the rostral segments, and the other 3 genes in the bithorax cluster coding for the more caudal segments.
Lastly, the homeotic transformations discussed were the antennapedia and bithorax transformations. It is important to remember that the antennapedia transformation results in a change from antenna to legs on the head of the Drosophila. This results in the Drosophila not having the sensory functions that it would originally have in the wild type. Second, the bithorax transformation results in a duplicate thorax. This causes the absence of the halteres that is normally seen in the wild-type fly, and instead a second pair of wings forms. This causes a little less balance for the fly while it is in flight. Thank you guys for watching our presentation. We hope you enjoyed.